We know that there are billions of chemical reactions happening inside your body. Although they happen at lightning speed inside cells when these reactions are run inside test tubes in the lab instead of a body. They happen at the snail's pace. What explains this difference in speed? What do our cells have that a test tube lacks? The answer is enzymes. Enzymes are life's greatest facilitators. They create the conditions needed for the biochemical reactions to happen fast. The general name that chemists use for the chemical entity that increase the speed of reaction is a catalyst. Enzymes are biological catalysts. They catalyze the chemical reactions that happens inside the living things. Now these are the contents of our today's topic. Number 1, chemistry. Number 2, classification. Number 3, mechanism of enzyme. Number 4, enzyme kinematics. Number 5, inhibition. Number 6, activation. And number 7, specificity. Enzymes are basically proteins that speed up the rate of chemical reaction in the living organism. An enzyme acts as catalyst for specific chemical reactions, converting the specific set of reactants called substrate into a specific product. Without enzyme, life as we know it would not exist. Introduction. In introduction, we are going to discuss enzymes are biological catalysts. They are 3D globular proteins and they are target specific in nature. Student, as we know that enzyme is a substance that acts as a catalyst in living organisms, regulating the rate at which chemical reactions proceeds without itself being altered in the process. Structure of enzymes. Students, as we know that enzymes are made up of protein, they are protein in nature. Actually, they are made up of long polypeptide chains that are folded on each other and forms a big protein molecule that has the catalytic activity. And this protein molecule, which we called an enzyme, has got two types of sites. One is the catalytic site and other one is the binding site. In this picture, you have seen the folded structure of protein molecule of enzyme that has two sides. One is which is labeled in the red one is the catalytic site and another one is the blue one which is labeled as the binding site. As far as the name shows, the catalytic site, it activates the chemical reactions and fasten up the chemical reaction, whereas the binding site is the site where the substrate is going to attach with the enzyme in order to proceed into the product. Now here is the further explanation of the active sites. Active sites can be further divided into number 1, binding site, number 2, catalytic site. Enzymes are target specific in nature. Enzymes obtain their specificity towards their substrates because of their structure. The protein structure is so designed in such a way that only the substrate that the enzymes are supposed to act upon undergo any change. The enzymes are specific because it allows control of the biochemical reactions without which the body would go haywire. As far as the specificity of enzyme is concerned, a few enzymes exhibits absolute specificity. That is, they will catalyze only one particular reaction. Other enzymes will be specific for a particular type of chemical bond or a functional group. That is, it in a broad way. In general, there are 4 distinct types of specificity related to the enzyme reaction and their target specific nature. The reactants used in a reaction are also known as substrate. These substrates require enzyme to be specific so that enzyme acts on the correct substrate or a bond to catalyze the desired reaction. More precisely, the specificity of an enzyme is due to the precise interaction of the substrate with the enzyme. Cofactors. A cofactor is a non-protein chemical compound or a metallic ion that is required for an enzyme's activity as a catalyst. a substance that increase the rate of a chemical reaction. Cofactors can be divided into many types. Here are the subdivisions of the cofactors. The cofactors majorly divided into two parts. Number one is the essential ions and number two are the coenzymes. The essential ions are further divided into two subcategories. One is the activator ions and other one is the metal ions. Whereas the coenzyme also divided into sub two categories. Number one is the co-substrate and second one is the prosthetic group. Coenzyme and cofactors are molecules that help an enzyme or protein to function appropriately. Coenzymes are organic molecules and quite often bind loosely to the active site of an enzyme and aid in substrate requirement, whereas cofactors do not bind the enzyme. Here in this picture, the coenzyme first attach with the enzyme and then the substrate is capable to attach with the enzyme in order to make the further reaction proceedings, that is the substrate will be converted to product. Cofactors are helper molecules and can be inorganic or organic in nature. These include metal ions and are often required to increase the rate of catalysis of a given reaction catalyzed by the specific enzyme. These enzymes and cofactors play an integral role in the number of cellular metabolism reactions, playing both structural and functional roles to aid in catalysis. Substrate. In chemistry, a substrate is typically the chemical species being observed in a chemical reaction, which reacts with a reagent to generate a product. It can also refer to a surface on which other chemical reactions are performed or play a supporting role in a variety of stereoscopic and microscopic techniques. Site of enzyme synthesis. Almost all enzymes are proteins. Like all proteins, they are produced by ribosomes, which are in the cytoplasm, the part of the cell that is outside the cell nucleus. After an enzyme is synthesized, it may be modified in the Golgi apparatus or elsewhere in the cytoplasm. As far as the polypeptide chains are concerned, a polypeptide chain which ultimately gives rise to the protein structure is in turn made up of amino acids. Therefore, to answer the question how enzymes are made, we need to understand how polypeptides are synthesized in the cell. Actually, protein synthesis hub is the ribosome. Wherever the ribosomes are present inside the cell, protein synthesis is there in that particular area. So enzymes are protein in nature and are made up of so many polypeptide chains, so they are synthesized in the ribosomes. Enzymes, as you already know, are the biological catalysts. Therefore, enzymes are synthesized by all living cells, including the lower animal cells or higher animal cells or prokaryotic or eukaryotic cells. They all are capable to synthesize the enzymes in their cytoplasmic structures or inside their cytoplasms. Enzyme classification is on the basis of the two types. Number one, on the basis of their secretion, and number two, on the basis of their mode of action. Types of enzymes on the basis of secretion. As far as types of enzymes on the basis of secretion is concerned, there are of two types. Number one is the intracellular enzymes. These are present in the cytoplasm, mitochondria and nucleus of the cell. Second one is the extracellular enzymes. These are the enzymes of the digestive tracts, that means lipase, protease and amylase. Types of enzymes on the basis of the types of reactions carried out by the enzymes. These are of 6 types. Number 1, oxidoreductase. Number 2, transferase. Number 3, hydrolases. Number 4, lyases. Number 5, isomerases. And number 6, ligases. Characteristics. As far as the characteristics of enzyme is concerned, every enzyme has 6 basic characteristics that are defined in this slide. Number 1, speed up the chemical reaction, that is enzymes are capable, all type of enzymes are capable to speed up the chemical reaction. Number 2, does not affect the nature of end product, that means it will not affect the chemical composition of the product that is being synthesized. Number 3, target specific in nature, that means they only target towards their specific substrate. Number 4, small amount needed for the bulk reaction. Number 5, sensitive to temperature, pH, substrate concentration. And number 6, work well with cofactor and coenzyme. The Enzyme Commission number is a numerical classification scheme for enzymes based on the chemical reactions they catalyzed. As a system of enzyme nomenclature, every Enzyme Commission number is associated with a recommended name for the respective enzyme. As far as the nomenclature of enzyme is concerned, enzymes are named according to the substrate catalyzed. Some were named before a systematic way of naming enzyme was formed. By adding suffix "ase" at the end of the name of the substrate, enzymes are named like ligase, protease, amylase, carbohydrase, and so on. Enzymes for catalyzing the hydrolysis is termed as hydrolase. Now there are some examples through which we can easily understand the nomenclature of the enzyme. Substrate, enzyme, product. Now the substrate is lactose, so the enzyme will be lactase, and the product which is formed by the reaction of the enzyme substrate is glucose and galactose. When the substrate is maltose, enzyme will be maltase and the product will be glucose. When the substrate is cellulose, enzyme will be cellulase and product will be glucose. When lipid is there, the enzyme will be lipase and product will be glycerol and fatty acid. And when the starch will be the substrate, enzyme will be amylase and product will be maltose. When proteins are there, enzymes will be protease and the products are peptides and polypeptides. Enzymes are responsible to lowering down the activation energy. Activation energy is the energy required to break the bonds of the substrate and convert it into the product. As enzymes are capable to lowering the activation energy by providing catalytic reaction to substrate, so substrate will be converted to product in short time period. Here in this graph, it is clearly shown that less activation energy is required when the enzyme is catalyzed the reaction in order to get the product. When there is no enzyme, the activation energy is high to break the bond of the substrate and to get the product. When the catalyst is added, that is enzyme is added, the activation energy level becomes low and the enzyme catalyzed pathway will become shorter, and in a short period of time product is achieved. The specific action of enzyme with a single substrate can be explained using a lock and key analogy first postulated in 1894 by Emil Fischer. In this analogy, the lock is the enzyme and the key is the substrate. Only the correctly sized key substrate fits into the keyholes active sites of the lock enzymes. Smaller keys, larger keys, or incorrectly positioned key teeth on the keys, incorrectly shaped or sized substrate molecules do not fit into the lock, that is enzyme. Only the correctly shaped keys open a particular lock. This is illustrated in the given picture also. Not all experimental evidences can be adequately explained by using the so-called rigid enzyme model assumed by the lock and key theory. For this reason, a modification called the Induced Fit Theory has been proposed. The Induced Fit Theory assumes that the substrate plays a role in determining the final shape of the enzyme and that the enzyme is particularly flexible. This explains why certain compounds can bind to the enzyme but do not react because the enzyme has been distorted too much. Other molecules may be too small to induce the proper alignment and therefore cannot react. Only the proper substrate is capable of inducing the proper alignment of the active sites. That is how the induced fit model works. Factors affecting the rate of enzyme catalyzed reaction. Basically, there are three factors that affects on the Rate of reaction. Number 1, temperature. Number 2, hydrogen ion concentration or pH. And number 3, substrate concentration. Like most chemical reaction, the rate of enzyme catalyzed reaction increases as the temperature is raised. The 10 degrees centigrade rises in temperature will increase the activity of most enzyme by 50 to 100%. Variation in the reaction temperature as small as 1 or 2 degree may introduce changes of 20 to 30% in the results. In the case of enzymatic reaction, this is complicated by the fact that many enzymes are adversely affected by high temperatures. This is shown in the figures too. The reaction rate increases with the temperature to a maximum level when abruptly decline with further increase of temperature because the most of the animal enzyme rapidly become denatured at the temperature above than 40 degrees centigrade. Over a period of time, enzymes will be deactivated at even moderate temperatures. Storage of enzyme at 5 degrees centigrade or below is generally the most suitable. Some enzymes loses their activity when they are frozen. Enzymes are affected by changes in pH. The most favorable pH is the value or the point where the enzyme is most active, and it is known as optimum pH. Extremely high or low pH values generally result in complete loss of activity for most of the enzyme. pH is also a factor in the stability of enzyme. As with activity for each enzyme, there is also a region of pH optimal stability. It has been experimentally observed that if the amount of the enzyme is kept constant and the substrate concentration is then gradually increased, the reaction velocity will increase until it reaches a maximum. After this point, increase in substrate concentration will not increase the velocity of the reaction. It is theorized that when the maximum velocity had been reached, all the available enzyme has been converted into enzyme substrate complex. This is the point on which the rate of reaction will not further increase until and unless all substrate will be detached from the enzymes and product is formed. Low substrate or high substrate concentration will directly affect the rate of enzyme activity. Now the summary of the lesson. Enzymes are protein molecules. Enzymes can be reused again. They speed up the chemical reaction. They lower the activation energy. And they are affected by temperature, pH and amount of substrate.